All right, in this lesson, we're going to talk about bacterial pathology and the treatment. So we know a little bit about their classification. Now we want to know what happens when they enter our body. All right, so many different ways in which you can get a bacterial infection. You can get it through food or water, really common. Food poisoning you're talking about. You're also talking about different examples where water supplies are contaminated. In Sydney many years ago, there was a contamination of the water supply. People had to boil their water or buy bottled water for a long period of time until it was cleared out. Fecal oral really common as well, mainly due to hygiene practices, often in children or people in developing nations where the possibility of clean water is minimal to start with, let alone used to wash hands. Direct contact, very common. We're not necessarily meaning we're just touching other people and therefore getting their bacteria. It's got to be about giving it an opportunity to jump into our system. So things like paper cuts, open wounds. Pro tip, don't Google open wound. It's a pretty horrible sight. And that's the way things jump in when our external defences are compromised. Water droplets, we're talking about that sneezing, coughing, flu season situation where we're coughing out bacteria and it's staying in water droplets in the air and it's able to be picked up by other people. Vectors are those particular organisms that carry the bacterial infection. So the bubonic plague back in the day was actually carried by fleas on rats, and therefore humans have contracted it from the fleas on the rats. So they had to go around killing a bunch of rats to hopefully get rid of the situation. There are also carriers, and that what a carrier is a person who is an asymptomatic carrier of the pathogen. They have the pathogen in them, they don't actually see any symptoms of it. Typhoid Mary is the most common case of this, and it's pretty interesting story, and it's a little bit funny at times in that Typhoid Mary was a person who immigrated to the US from Ireland, and she was a cook, and she worked for many different families, and every single one of those families kept developing fevers and diarrhea, and it was more of a case than just simple food poisoning. It was a real repeated thing. And if you have a look at this, the newspaper ran a bit of a story on it, and she's cracking skulls into a pan like eggs. So the most harmless and yet the most dangerous woman in America. The typhoid fever is caused by a bacteria called Salmonella typhi. So we've heard of Salmonella, and that generally happens when you hear about it when you're talking about food poisoning, but this particular one is a little bit more drastic. It does cause vomiting, diarrhea, but it also leads to those fevers and headaches, and a little bit more problematic, particularly back in the 1900s where treatment wasn't as readily available as it is now. Typhoid Mary was isolated. They tracked her down. They isolated her and told her, you're not allowed to be cook anymore. She went out to become a laundry worker, and then she just went, I'm going to change my name and be cook again, and pretty much pattern of sickness all along the US, and they couldn't really ever find her. When they did, they isolated her, and I think she spent the last something like 23 years of her life in isolation, only cooking for herself. Right, bacteria evades hosts using molecular mimicry, which means they can actually make themselves look like things in our body. So this picture explains it really well. If this is the microbe here, and what the antibodies are looking for is a particular pattern, and usually they'd be able to do that, they actually make themselves look like something that we have in our cells. So a protein that we have, and it mimics the shape of that protein. Now, the flip side of this is that these microbes can go undetected, but the opposite end of that is that our body can actually start an immune reaction on self proteins. And we're talking about allergic reactions there. There's also quite a number of diseases like the Crohn's disease and a few others like where your body is attacking itself. Rheumatoid arthritis is another one. I get eczema, and that's an example of it as well, just an overreaction of the immune system. All right, bacteria can also target immune cells to suppress the antibodies being produced. So rather than go after any other cell, they go after the immune cells that would usually be stopping it. And they can also colonize inside those ones. So even though phagocytes are there to engulf them and break them down, they can actually develop defenses to stay alive and therefore live inside the phagocyte, which is pretty clever. Latency is interesting because it means that bacteria can stay in the system and then not actually cause pathology until they feel like it's an appropriate time, often when the person is immunocompromised. So when there's an opening available and the immune system is down a little bit, that's when bacteria go, yep, this is our chance. Bacteria cause pathology with toxins, right? They can degrade tissue to enter the wounds. And there's endotoxins and exotoxins. And those exotoxins are the ones that are excreted, right, using exocytosis, hence exotoxins. However, endotoxins are part of the cell itself. And when the phagocyte actually engulfs this one, what will happen is these toxins will come off as the bacteria starts to break down, and therefore also killing the immune cells. So it's a little bit of a last-ditch defense mechanism. They have fimbriae, or little hair-like projections to attach to the mucous membrane. Now, any opening in our body, our mouth, our nose, our ears, every other opening is known as a mucous membrane, and they're sticky. And what that usually does is expel, or give the opportunity for the body to expel things like bacteria. But bacteria also have these little hair-like projections around their whole length in order to grip on, so they don't get washed away as easily. Now, these are a little bit different to, say, cilia. They aren't there for motility, they aren't there for movement, they are there to simply stick on and keep themselves attached. Bacteria also have enzymes to break down our cage of fibrin and connective tissues so that they can start to break down the tissue and therefore get further into the body. And really all bacteria are trying to do is find a nice, squishy, moist, nutrient-filled home. And what we're trying to do is prevent them from that home being us. And when we start talking about pathology, we're talking about, right, what are these things actually doing to us? And that's the important thing. Now, these toxins can do a whole heap of stuff. Inhibit protein synthesis, damage cell membranes, stop nerve functions. We've talked about the endo and exotoxins. Now, botulinum toxin is actually used as Botox. It's injected into the face, and what it does is it stops muscle contraction. So it stops the movement of all our face, and therefore stops the potential for wrinkles to happen. Now, if you're getting the Botox injected every now and then, it has to keep being pushed in because our body can become a little bit immune to it. If you have a look here, this is our normal. We know this is a synapse, and the the neurotransmitters are pushed out and then pushed out onto the receptors, and then the muscle can do the contraction. However, botulinum toxin it stops these neurotransmitters from being released and therefore stops contraction of muscle. The opposite's happening in the tetanus toxin in that it's actually stopping the stopping of the contraction. So it's only allowing the contraction over and over and over and not allowing the relaxation of the muscle. So while the Botox allows for relaxation, the tetanus toxin only allows for contraction. So that's why there's that rigidity of the muscles during tetanus. All right, this one's a little bit gross, and one of the enzymes that one particular bacteria uses, Clostridium perfringens, uses an enzyme to break down our tissue. So what happens if we have lack of blood supply to a particular tissue, which can sometimes happen in diabetes, for example, your circulation is no good, then the bacteria can jump on board and it can start to break down our skin and therefore get deeper and deeper into our system. So this is a not so great picture of gangrene of some toes, and this is a diabetic case where the circulation is very, very poor and the skin starts to break down, the tissue starts to die off, and therefore the bacteria can jump on in. We do have treatment though. We do have antibiotics, and that's fantastic because once upon a time they didn't have those. They are naturally occurring compounds, and they can be broad or narrow spectrum. And broad means they're covering off and trying to kill as much as they can. Spectrum means they are specifically tailored to kill a particular bacteria. They are semi-synthetic, so sometimes we can now make those naturally occurring compounds and we can mix them with different things, often things from fungi and other bacteria as well to help us out. They are also, once upon a time, and in some countries still are, used for livestock. They encourage growth. They don't encourage stress, sorry, they encourage growth during stress. So if you have, say, in the situations where we're talking live exports, we have a lot of animals pushed in together, it does allow for them to grow despite their surrounds, I guess. And it's leading to resistance, and that is not a good thing. We have categories of antibiotics, and they can target, say, breaking down the cell wall. They can break protein synthesis or just stop it from happening. They can stop DNA duplication and therefore stop replication of that DNA, the bacteria, and therefore slow down their colony growth. Or we can actually break down some of the things or stop the synthesis of things that they are trying to make for their own metabolism. The history of antibiotics is pretty interesting. This guy's Alexander Fleming, and he is kind of references the discoverer of penicillin. And what he did was He was previously an army doctor, and he had a lot of mold growing on his agar plates that he was trying to grow bacteria on, and he found that wherever the mold was growing, the bacteria wasn't. So he noted it down. He decided to follow up. He did some follow up. He didn't really get great results. He said, all right, this could be used as a disinfectant, and then he left it for years and years and years. So along comes Howard Florey, and Howard Florey is an Australian, and he's living in Oxford at the time, and he's read Fleming's paper, and he goes, I reckon I can do something with this. So he starts to use it on mice, and he's injected mice with a particular disease, and he's used the control group, given them nothing, and the other control group, given them the penicillin injection. Now, not that many, it's not a big sample size here, but the 4 that didn't have any penicillin that died, the 4 that did all survived, right? So he's gone, this is amazing, let's do something with this, wrote it up. Fleming read about it, he went and visited his lab. It seemed like, oh yeah, guess that's okay, seems interesting, whatever, walked away. Many years later, Fleming's got a friend who's dying, and he's called up Florey and he said, can I use some of your bacteria or your mold isolate, please, to use as penicillin to see if I can save my friend? His friend lives, and both of them end up writing it all up, and him and Florey's lab assistant as well, they get a Nobel Prize in 1945 for medicine. So quite an interesting story, and Australia likes to claim our important people, and Florey was once upon a time on the old $50 note. All right. Now, because there is such massive use of antibiotics in the world, it does lead to resistance. Okay, one of these two out of the big bunch, most of these are going to die off because if the antibiotic's going to attack them, if they are bacteria. However, there are a couple that will have a little mutant resistant strain, and what will happen is they will go on, they will breed, and suddenly our bacteria are resistant to the antibiotics that we have. We have MRSA, which used to be called methicillin-resistant Staph aureus, but is now called multiple resistant Staph aureus because we've hit it with more and more drugs, and we've found it, unfortunately, it's resistant to a number of different antibiotics, not just methicillin. Pretty gross skin rash, really, very, very dangerous, and it's often hospital acquired. Okay, those really sick people end up in the hospital where lots and lots of antibiotics are used, and that is exactly where those strains become very, very prominent.